And we've started. Okay, so pick up where we left. Today's important because we really talk about resting membrane potential, and it's extraordinarily important, as well as the utilization of active transport mechanisms. And I think that's important as well. So, get back to where and there really is not a lot to say in that first PowerPoint. Like I said, and when we get to the third portion of it, which is all about parts of the cell, I mean, I'll laundry list that relatively quickly, I am sure. But we left here with osmosis, and the whole idea is because our cells don't have the cell wall to deal with shifts of water. Water is particularly important to understand the ramifications, as it were, of not enough or too much. And it's all about tonicity and the whole language. So again, in this class, from a testing perspective, we use the term solute and solvent. Solvent, unless it's characterized otherwise, indicates water. And solutions, by definition, are things dissolved in water. Okay. When you hear it scientifically, something about a tincture means it's at least half alcohol. That's the distinction between. In the old days, we used to have tincture of iodine and tincture of methylate that were used as topical antiseptics, and a variety of reasons why that's not the case anymore. For that, I'll send you one of my micro lectures when we do that part. Okay. So isotonic media is what we try to use physiologically when we don't want to induce either not enough or too much water. We don't want to pull water out or push water in. Sometimes you do. If someone's dehydrated, you might want to push water in. If somebody's overhydrated, you might want to pull it out. And typically we use isotonic media based on, again, the colligative properties, which effectively is freezing point, because everything you add as a solvent, as a solute rather, to a solution will lower its freezing point. So hypertonic induces crenation. We do the in lab, and you may have done it. You do it with red cells, and you can see the red cells kind of square up and go from the round discs. And if you try to do it the other way with like pure water, effectively deionized water, you almost can't do it on the microscope. And I'm fast focusing a microscope. And I never get to see those cells swell. The only way I think you can see it is if you're having something you're visualizing in real time. So that's really the part of it that's important, and they go through the whole, and that's chemistry. We're not going to do molarity, V-molality. Just understand that that's what isotonic media, the most common ones that are isotonic. Normal saline, 0.85%. Saline, D5W, 5% dextrose, or 5 grams of dextrose dissolved in 100 milliliters of water. So it's, and sort of like, you want to think of intravenous Gatorade, which is got a combination of solutes that are sort of osmotically balanced and desirable. There's something called lactated Ringer's solution, and we have used a lot of those. In my years in hospital treatments, most time we infuse something that was in normal saline, but I every so often use it for different things. And that's the experiment, the classic experiment, where you can see the crenation of the red cell. Again, if you can't capture this in real time, they pop on you. And there's a classic biconcave disc. Then we do a very long and I think a very interesting unit on the blood, one of my favorites to do. So not doing that. Okay, so we finally finished that bad boy. Move on to the next one, which is B. And B's pretty chock full of goodies, because everything we have talked about so far has been passive. And in living systems, we like passive transport. You don't want to expend energy needlessly. Believe me, that's my mantra. I'm indolent. You know what indolent is? Indolent makes lazy look active. I'd rather sit than stand. I'd rather lie down than sit. Just the way it is. There's one of the late actor Robert Morley did a wonderful essay of that years ago. And none of you would know who I'm talking about, would you? Not in the least. Did you ever watch an old movie in your life?
No.
There you go.
I'm not good at movies. If it makes you feel any better, I just watched The Lion King for the first time.
I feel better.
Like last semester.
I feel better.
Yeah.
The music is brilliant.
We didn't even watch it the whole way through, but I watched like 80% of it.
I feel better. That was Elton John did the music, and Sir Tim. Trying what?
Was it Phil Collins?
No, Tim something did. Phil Collins might have been doing some of the music in it, but and this other guy did the lyrics because Elton John was always did music, was never fancied himself as a lyricist. And all the great songs he did the music, and his writing partners the lyrics. Yes, good for you.
Thank you.
I've had that. I would not consider that old, but it's okay.
Well, maybe when I actually watch it the whole way through, I'll start watching your old movies. But it took me 21 years to watch The Lion King, so it's not really looking good.
I hear you. Maybe I should just show scenes of these 1930s movies.
There you go.
If nothing else, for the sword fights.
You can put it on during lab.
It would probably be a good thing. Okay, so when it comes to expending energy, we do it limitedly, and when we do it, we try to do extra, if you want to think of it that way. So active transport and vesicular transport, by definition, when you see those terms, it means you have to expend a certain amount of the ATP that you have. So whether it's things that are too large for the channels, not lipid soluble, not, you're going against the concentration gradient, it doesn't matter. And again, carrier proteins. And this is the interesting part, when we have this wonderful ability to use the same energy to push something in and pull something in. And we see this a lot. And you'll see a lot of classic examples of sodium potassium pumps effectively do this. And so that you're basically pumping things out and pulling stuff back in. So anti-porters, anti going in an opposite direction, versus sim porters using almost like drafting that you would if you were swimming or cycling or racing and coming through. So you're using that flow that you created to ease their way in for other particles. Okay, and so those are really good examples. Most of the time when we move sodium around its active transport, you'll see a particularly good examples of this in the kidney, and they do a lot, and that's the primary active transport. And utilizing that sodium and the gradients that it creates, we do a lot of moving, like for instance, glucose will move with sodium. You use different compounds in the kidney to do. So that's the secondary. So the required energy is obtained indirectly, say from the typical sodium gradients that are part of the primary active transport. So again, you're optimizing the utilization of your energy. And I think, and that's, to me, there's always a limited amount. And living systems, you try to dedicate that. At certain times, you always wonder, particularly as you get older, and and friends and family are getting older, and many of them, unfortunately, are dying. You have a certain limited amount of resources. You can only fight so many battles. We've been saying this for a while. I mean, when an older person gets sick, maybe they can recover once or twice, but about the third go around, all those resources have been spent. There's only so much that they can do. So it makes you wonder. We used to call it total system failure along with it. So energy obtained from hydrolysis of ATP will be required to change the shape of those transport proteins. And so the classic examples, and anytime you use the word pump, it implies active transporters, I'm sure you know. Calcium, proton pumps, sodium, potassium. By far and away, the most abundant of these are the sodium potassium pumps. Nearly any cell that does anything has them. Particularly, and approximately half of our energy goes to these sodium potassium pumps, resetting rapidly neural, and muscular membranes. So that's the vast majority of your energy. When you hear about chronic fatigue, there's a lot of theories that chronic fatigue individuals, and in some people it may or may not be real. I'm not trying to dispute its presence, but just understand that we think that there are faults with regard to the mechanics of these pumps and their ability to, more appropriately or easily utilize energy. So it's the one that's most studied. They call it sodium potassium ATPase. And it basically, when nerve and muscle cells are busy, as they always are, we have sodium rushing into the cell and potassium rushing out. And we rapidly have to reset that because that's part of restoring the resting membrane potential, and moreover, restoring the balance of sodium, which should be more abundant outside the cell, and potassium more abundant inside the cell. So all plasma membranes have these, but the ones that are particularly excitable cells, nerve and muscle cells. The membrane, what happens is it's a combination of not just these channels, but these are these leakage channels, by definition constantly open and leaking, if you will, bits of sodium into the cell and potassium out of the cell. Remember the construct. There's always more potassium inside of the cell, always more sodium outside of the cell. And if there's not, those pumps get busy to recreate that. So the sodium potassium pumps antiport put the sodium out of the cell and potassium back in against the concentration gradient. And they give you, and you can look through these, and they have these individually. And the take home point for this is the 3-to-2 relationship. 3 sodiums, 2 potassiums. All you have to do is look up there on the periodic table, or in our case, the imaginary pediatric table. That's up there, and you know what's all in that first column, lithium, sodium, potassium. They all want to lose an electron in the worst way. Well, go away. I don't want you. Leave the house. Never dirty my towels again. That's a line from Duck Soup, which was 1934, with Marx Brothers. I'm not surprised. It's nearly 100 years old. But you can watch that on, do you have Cinemax or Max, we call it now?
Yeah.
You can watch any of those on TC, on Turner Classic Movies. On that point.
You have to make me a list.
I will.
Maybe I'll watch
I will. You'll see the distinction between modern humor, where we're fairly vulgar, versus,
Yeah, but I like vulgar. I think it's funny. So,
I'm a stranger in a strange land.
just saying.
There's a great line in that at the end of the movie where Groucho says to Margaret Dumont, who always paid the dowager, meaning this wealthy woman whose husband had died, he was always courting her for money. He goes, "We're fighting for your honor," which is more than you ever did. And just the way that he would be able to say those things. One of the really quick-witted people of all time. Nice Jewish boy from the Lower East Side in New York City with 4 other brothers. Just saying. Moving on. There actually was a Broadway show about them called Minnie's Boys. But she was the vaudevillian. Nevertheless, so because sodium, think of it this way, is smaller molecularly than potassium, roughly 3 sodiums and 2 potassiums are approximately the same size. So we constantly see this 3 being pumped in, 3 being pumped out, 2 being pumped back in. And you're going to see the same thing in the opposite direction when we get to neuromuscular physiology. When acetylcholine hits the motor end plate in a nerve, what effectively it does is it allows 3 sodiums to enter the cell, 2 potassiums to exit the cell, and it elevates the resting membrane potential to what we call threshold, and that generates an action potential. So they're just showing you through these slides, and I'm not going to go through them with you, other than to say that there they are. One thing about this textbook, what I like the best, and even if we switched off of this textbook and I, for whatever reason, still want to teach, I would use these. Making me look bad. I've played before.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
And so even in active cells, comparatively speaking, just to maintain the gradient, there's always some sodium-potassium pump activity. It's really what they're underscoring there. And it really is an important measure because the changes are dramatic in excitable cells like nerve and muscle cells that we'll get into in detail. And that's a lot of really what the first third of this course is really dealing with a lot of that activity. Okay. So depending on the gradient, again, we can have different kinds of secondary. Okay. Low sodium maintained inside the cell by the pumps. Sodium as it flows, okay, into the cell, again, may generate symporters with various things coming into the cell as well. So whenever the primary mechanism brings along others to the party, either coming in or going out, now we have secondary active transport. So it doesn't really matter what it is. They're depicting glucose here because you can see that hexagonal shape. And they're all going to be, comparatively speaking, small molecules. Okay, good? Okay. The other way, and we do a ton of it, because most of the stuff, when we move bulk, think of it, the distinction is, think of all the boxes you get delivered to you from Amazon, or, as opposed to maybe tiny items in a mailbox. So when it's a little bit easier to throw something in the mailbox, whereas if it's our house, they're bringing boxes like crazy because we're going to repackage them and resell them at an obscene profit. I just listed a Star Wars Saga chess set with board and there are no new ones around. It's about a good 25 years old for $150. What? 25. How much to shove it? Probably make better for $100. Well, then you have to share the same with your rich uncle in Washington. Remember him? Good old Uncle Sam. Bane of my existence. That's what the next month and a half are about. Compiling tax records. No. It's just P&L, profit and loss sheet. My wife's business is sole proprietorship. So you just add up what you made and what all your expenses are, and that's your number. So because she effectively she's self-employed after working as a nurse for many years. It's okay. I still work for a living. I double dip. They pay me here to talk. What could be better? And I get Social Security. I double dip. What a country. Okay, so when we're bringing large stuff, when you're getting a truckload delivered, vesicular transport. And so membrane-bound organelles, and you've all done the cell 1,000 times, so I'm not going to spend a lot of time on it. You know, all along that endoplasmic reticulum, from rough to smooth to Golgi bodies to out we go. Okay, this is how they're moved. And every one of those steps requires energy. All those motor proteins that are carting vesicles, effectively membrane-bound containers with product to ship. Okay, and that's what's going on. And so that, again, mostly by way of ATP. And it doesn't matter what direction, whether you're bringing it in or pushing it out. So you can either bring it in, endocytosis, or you can push it out, exocytosis. And we do it constantly. Our lysosomes are as busy as anything else. Every little particle that you ingest, as you well know, that comes in by that bulk method, enters by way of a lysosome, enters by way of an endosome, fuses with a lysosome, is digested or destroyed depending on whether it's beneficial or not, and then you have to get rid of the garbage. That's basically how it works, continuously. It's very straightforward. So you can have phagocytosis, particles, pinocytosis, liquids or oils, and very frequently receptor-mediated endocytosis. So that's when it targets, when there's specific receptors in the membrane that when they are latch on to the particular group of particle or particles will form that vesicle and facilitate the ingestion by way of endocytosis. That's a really important mechanism. Okay? You want to think of it this way in simplistic terms. Simple diffusion is usually associated with hydrophobic particles, gases, oxygen and carbon dioxide exchange. Rapid doesn't require, it's just thin membrane with two barriers, one on either side, that are simple squamous epithelia with very little in between them. Facilitated diffusion indirectly has to do with a molecule opening that channel. And that typically would be associated with insulin, which does more than just glucose, but one of its major activities is to facilitate the entry of glucose into the cell. When you hear about people with hereditary lipidemias, meaning they have high cholesterol and other fat levels, and they have it from when they're comparatively very young and it's a familial tendency, a lot of times what they're missing is they don't have the receptors or a sufficient amount of receptors to allow cholesterol and cholesterol-like molecules to be brought into the cell to be processed. So where do they stay? In the bloodstream, where they cause trouble. You want to think of it that way. So that's where, so it's not always diet and exercise. Some folks benefit by certain medications that are designed to facilitate the entry or augment the entry of these things. It's pretty hard to reprogram the cells that are there. And exocytosis is effectively spitting out. Transcytosis happens digestively a lot because the particles, even though they're brought into the cell, they really don't interact with it. They just occupy a vesicle that's brought all the way through and exits. There's no interaction with a lysosome per se, right? So endocytosis, protein-coated vesicles, typically receptors, unique receptors. This is a big problem. Pathogens hijacking it. Now, again, I know you didn't talk about it a lot in micro, but this is one of those things, like where HIV can hijack that mechanism, where tuberculosis can hijack that mechanism. You have to understand that there are certain, and it allows them to multiply within a living cell before they start to alter the markers immunologically outside. So it's there's a lot of complexity to this particular process. So it's things that as we begin to understand what we call the pathogenesis, the disease-causing mechanisms of these things that go on. And then lysosome and undergo. So they can one way or the other. They're either going to fuse, and make that whatever you want to call it, a phagosome, and then an endolysosome and gobble it up and spit out the contents, or it can just go straight through without fusing with it, if you want, without doing more with this particular illustration. The oddball thing, and maybe you covered this or not with genetics, is there are certain disorders called lysosomal storage disorders. They lose the mechanism to expel breakdown products, and they accumulate within the cell and damage it. By far and away, the most famous of these is an autosomal recessive disease prominent in Ashkenazi Jewish families called Tay-Sachs disease, which you probably covered. My first wife and I, at the time when we did genetic counseling, the only blood test that was available was for Tay-Sachs. Since we were both of Ashkenazi Jewish lineage, which means European Jewish rather than Middle Eastern Jewish lineage, we both tested. Neither one of us were carriers. So it was non-starter. There were other factors why we couldn't have kids biologically. And then we foolishly adopted 3 of them. What were we thinking? Anybody want to go to the World of Wheels down there in the convention center? There's cars on the 2nd floor. Make him an offer he can't refuse. Okay, moving on. So, whatever you want to call it, phagosomes, amoeboid motion, and that's basically how they sort of scooch around, change shape. Without getting into the ins and outs, you probably covered a lot of that in cell and molecular. Basically, the small microfilaments being able to manipulate shape change and create motion associated with it. Here you can see they'd be forming these sort of little arms and pull in material, often triggered by receptors. And the same thing here, whether it's liquid or whether it's solid. Here, typically we see clathrin-coated pits. And that's what I meant. Low density lipoproteins. And unfortunately, this is where the problems are. Viruses, diphtheria, certain toxins of diphtheria and cholera, which are toxin based. Even these, and we'll learn about when we talk about smooth muscles, caveolae, okay, which are different types, which have different kind of coatings, but very, very specific. And you can see tetanus can hijack that mechanism. So there's a lot that goes on. I mean, you can delve very deeply into these, and we'll look at some of the ones that are there. And exocytosis just reverses it. Usually, again, various signals or changes in voltage activity. They're secretory vesicles by definition. When we get to neuromuscular physiology, the release of whatever the neurotransmitter is, whether it's triggering a muscle to fire or whether it is triggering a nerve to fire, by definition, those are exocytosis process. And those vesicles, okay, all are secretory vesicles. They're filled with either granules or liquid or whatever molecular basis of the common transmitters, choline, serotonin, dopamine, whatever it happens to be. And I'm sure you covered V snares and T snares, correct? Okay. So whether it's on the membrane itself or it's on the vesicle itself, that triggers the process that are there, all the different things, whether it's simple as mucus in the gut or in the respiratory system. And they show the interesting thing when you get to the snare process, and here it's a pretty good slide series to show how these appendages, come together, the V and T snares, and then facilitate that. What very powerful chemical toxin blocks that activity for acetylcholine at the neuromuscular junction? Pardon me? What particular toxin, biological toxin, blocks that process of being able to excrete acetylcholine at the neuromuscular junction, famously.
I have no idea.
It's a paralyzing agent. And is sometimes used, here's the hint, cosmetically to treat wrinkles.
Oh God.
There you go.
Oh God.
Somebody wants in, I think. Anybody want in out there? No, you're all good. Just saying. So it's Botox. That's right.
They might have commented.
Hmm?
Yeah, like check the chat.
Oh, where's a chat? This
Right there.
botulism. You're correct. See, we have a winner in New York. Well done. Okay, so botulism toxin or botulinum is, and it may be the strongest of all the toxins, and that's why it's a neurotoxin. We covered this just like tetanus. I beg your pardon. Just like, let me get through this and get back to where we were. Just all right. I'm presenting. I know that. Stop it. Driving me with sugar. Sugar. So, and what it does is it blocks that process. It's very, very interesting. So we're using it a lot for everything from headaches. It has a lifespan of around 6 months, things like that. My daughter was an esthetician. She still is, when she was doing it at one of these places where she was more into sales and things like that, not so much doing it, even though she was trained to do it. And here we are again with the resting membrane potential that's so terribly important. And again, it's right across the membrane. The internal cellular milieu, the intracellular fluid, and the extracellular fluid are not charged. The charge is merely distributed immediately across the membrane. And that's also important, and that's where we get the term polarity, because of being the opposite nature of their charges that are there. And that becomes really important. This was the key, and the key player is potassium. I say this all the time. Doctors, when you see someone's potassium start to go in the wrong direction, typically low, that is a big deal. Sodium varies enough. There's a wide range. Normals for sodium, about 125 to 145. So if it's 140 or 127, nobody gets excited. Potassium, when it goes low, people get worried, because what happens, it creates a lot of instability, particularly cardiovascular. And when we start looking at electrocardiography, you're going to see that that's a big player that's involved. And take it from somebody who got intravenous potassium. It ain't fun. Ain't fun. Okay, so that's part of it, as well as there. So the potassium effectively is more of the driving force, and really the resting membrane potential. Potassium would be minus 90. The reattraction of sodium, because of the electrical gradient, and although the present, although less so, sodium channels, leak channels, allows that to be reabsorbed, blah, 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 blah. And again, you have things that negate it. What negates the positivity on either side, proteins which accumulated in the side of the cell have a negative charge, and chloride ions on the outside. You're going to see chlorides at play here too when we relatively soon when we start getting to neurotransmitters. Chloride gated channels are how, for instance, impacting them is one of the ways alcohol works. Why it's sort of a sedative in its own way. And they're just showing, here's the proteins with a negative charge on the inside, the chloride on the outside, balancing out the charge, which you would think would be positive because of the abundance of positive particles, but it's not. Then here they go, the transport and resting membrane, which brings us to this aspect of it, these environmental interactions, which are more important than you think. A lot of people, we give them a short shift, okay? And they involve areas of the glycocalyx, being, again, those sugars that are adherent to the proteins. It involves membrane receptors and these adhesion molecules. So you see, as we laundry list through them, you'll see it. So there's tons of these, just immense amounts. So again, very few cells function independently. Lining cells are adherent to one another. Again, probably other than white blood cells, everything else works as a unit. Red blood cells are an oddball because it's their aggregate effects. But all the rest of them are tethered to one another, either directly, like in the typical epithelial or lining cells, or in connective tissue cells. The very name tells you connected by a variety of fibers, most frequently collagen. And yeah, they play a role in those motor proteins, moving those vesicles along so they can move things. There's those molecules are part of what sort of interacts with various chemicals. When we get to inflammation, when you injure yourself, prick your finger, stub your toe, twist your ankle, cells inevitably are disrupted. They release chemicals. Those chemicals are signaling agents we call cytokines. Those chemicals induce other cells, particularly white blood cells, to come to the party to fix the problem and initiate the response that we collectively call inflammation. So it doesn't necessarily happen right away. In sport, a lot of times it'll happen later that day or the next day. So that's why you always pack people. You see on the sidelines with ice wrapped around them with compression, trying to limit the spread of those chemicals and to the limited degree of deformity because it's easier to fix it down the road from a sport medicine standpoint. So they're important. Okay. Additionally, a variety of things recreating the membrane junctions, transmitting signals. You'll see all of them. You've gone through this a lot previously. So binding sites for different cases. So contact is so interesting. Cells that touch and recognize each other. And again, it's what I said before. It's all about that signature of glycoproteins that are there. That's that self versus non-self. And it only takes a subtle change. So if something, again, if something's like this and there's just a tiny little change where something protrudes a little bit more or a little irregularly, immediately it sets up a chain reaction. So part of this is normal development and the idea of recognition of self versus non-self. It's also, and maybe you've seen this in the laboratory, where when you have cell cultures, they grow, and when they stop growing at a level, okay, they'll create a layer of cells on top of whatever the liquid is. You won't see irregularities or humps and bumps. In bacteria, there's certain morphological manifestations on a plate that helps you identify in the micro lab what kind of organism it is. They aggravate, they behave a little bit differently. But our kind of cells they fill the gap, and once it's all filled and they're all touching one another, it's called contact inhibition. They stop growing. In cancer, they lose that capability. And that's why you see irregular growths and invasive processes. That's one of the things that trigger us when we either look microscopically or macroscopically in surgery. And I've seen some cancers that have been pulled up, they are ugly. They're scary. They're either all mushy or messy, or they have little tentacles. And I mean, they are scary looking. Bad looking. And I remember seeing those, and the scarier they look, the worse the prognosis is for that individual. I mean, I haven't done any of that in 50 years, more or less, 40 or 50 years, but cancers are still cancers. Okay. And we know there's a tremendous amount of chemical signaling that goes on, triggering literally everything. And we'll go into what they do, whether it's neurotransmitters, whether it's hormones from the endocrine system, or these neat things vis-a-vis the inflammation called paracrines. And they play a significant role. And many of us inhibit them all the time to block inflammation if we take an Advil, we take an aspirin. Most of the way they work is they inhibit either the activation of or the activity of certain of these paracrine compounds that we collectively refer to as prostaglandins, because they were first identified in prostate secretions. I'm sure Dr. Hilwick told you about essential fatty acids. We know about essential amino acids, okay, and you have essential fatty acids. Arachidonic acid, for instance, something like that. And what they do is they fold like a hairpin, and that activates them. You know what they're abundant in? I had some earlier this morning. Peanut butter.
So a lot of peanut butter good?
But
A lot of peanut butter.
it's always good. It's a good thing.
Okay, but like for your health, is it good?
Oh, absolutely.
Okay, great.
Absolutely. We got a great, my wife found a great recipe for, are you ready for like a flour-free, all it is sugar, peanut butter, and an egg. And you're going to make the best peanut butter cookies you ever. Oh, they're great.
Really?
Like for gluten-free type stuff. You don't put any flour in them. Oh my God, they're great. So we go to Costco or to Sam's, those big double jars of Jif. Oh yeah, happy days. It's a food moment, everybody out there in virtual land. What could be better? Okay, so we collectively, when we use the term ligand, they are chemical signals. So you're going to hear a lot about ligand-gated channels. That would be an example of the neuromuscular junction and acetylcholine. Or voltage-gated channels, those are rapid alterations based on an action potential of membranes. Okay, and there's all sorts of different ways. Some work very fast. When ligands, a chemical, bonds at a neuromuscular junction, it allows sodium and potassium to move in opposite direction. Those are ion channels that are open and closed. They're very rapid. Others are much slower. Okay, and then this gives you back, and I'm not, and we could do this 1,000 times, and I'm not going to. The two basic ways, G proteins for water soluble and large molecules and proteins versus the fat soluble signaling that goes on. So if you've all seen this G protein thing in whatever form, whatever text or PowerPoint sequence it has. So G proteins, it's a chain reaction. The big G, as you all should know, like the big A for ATP, is from adenine, and therefore adenosine when it bonds with ribose. G is because they're guanine. So adenine triphosphate or adenosine triphosphate and guanosine triphosphate both have that energy-based capabilities of high energy molecules in their structures. Okay, and they can do anything. Ion channels, other enzymes, a variety of second messengers, typically either cyclic AMP. So we get a lot of mileage out of adenosine. So we have adenosine triphosphate, we lop off a phosphate, we have adenosine diphosphate, and basically they constantly ping pong that inorganic phosphate with one another. And I say this all the time, for you and I, we create and break down approximately every day an equal amount of ATP to ADP back and forth as what we weigh. How's that? Maybe the most fascinating molecule in the body. Just keep ping ponging. I mean, so all those channels that you saw, every cell with those sodium potassium pumps, what are they doing? Same thing, ATP, ADP, like that. And then calcium plays a role in this as well. And you've all seen this in some way, shape, or form. This sort of, it's like, the relay race model. Here's the ligand, call it insulin, if you will, targets a receptor on the outside, triggers the chain reaction based on a G protein or guanosine-based protein that becomes activated and then eventually works through a series to activate something else, some effector protein. We'll look at it in blood. We'll see adenylate cyclase, all those things. And then typically the chain reaction, famously the one that's best understood is the tyrosine kinase one. It's associated with insulin and diabetics, so it's just understood better. So if you want to review that, review that. How'd you like that?
That's great.
You're just saying that.
No, I'm being so honest, Dr. Stein. It was wonderful.
See, would you find this review useful? Okay. You see, this is Stein's simple view of how the human body works. Which brings us to the end. If you think we went fast through this, just wait. The only one we're going to take our time with is the cell cycle. Prophase, metaphase, anaphase, telophase, cytokinesis. Or as I like to call it, PMAT. It's because my wife's name is Patty and we call her mom P-Mom. It's easier. So This is the cell particles. What you've done this 1,000 times. So you have a sol gel, areas that are gel-like, areas that are solution-like, that make cytosol because of all this interesting milieu or combination, various insoluble molecules, depending on what kind of a cell it is, whether they're pigments, whether they're fatty droplets, whether they're glycogen, and a whole host of organelles. Some that are membrane bound and some that are not. Classically, the best example would be the ribosome, okay, being non-membrane bound and occasionally being tethered to the endoplasmic reticulum to create rough endoplasmic reticulum based on whatever that signaling molecule is that Dr. Hillwood talks to you guys about. The one that tells you we're going to the membrane to make a protein rather than just making a protein for inside the cell, right? Whatever that was. Those like ticks and tox. Boy, you really retain this information for the long haul. Nobody cares. I remember all this stuff, even stuff I'm not supposed to remember. If you really want, it's upsetting. I'll talk to my buddy who I've known for the better part of 60 years. And I'll go, remember when we did this? I have no idea what the hell you're talking. Remember we almost got arrested during that? No, I don't. Just saying. And I remember it like it was yesterday. You know, what people wore, what they had to eat. So I can't help it. Okay, and membrane bound, quick review, not organelles. Yeah, I know. And I like to say it's like pathognomonic.
Like being different. I know.
Not different. You know, it's like phlegmatic, phlegm. You know, diaphragm. If I remember how to pronounce it, then I know how to spell it.
Okay.
All those guys that are there, all that. The interesting ones to me are the ones that do interesting stuff. What's interesting about a mitochondria?
Powerhouse of brain cells.
Yeah. But what's unique about that? What's it got that no other organelle has other than one? Not a nucleus. It's got its own DNA. What kind of DNA? What? What? It has prokaryotic DNA. Yes, it's called. Didn't you learn about endosymbiotic theory? Do you know what I'm talking about? You don't know what I'm talking about. Endosymbiotic theory. You're terrible. The theory is that prokarya were gobbled up by an organism, and then when they were found to be useful, they were incorporated. So bacterial DNA has, just come on in, don't fret. Bacterial DNA, it's okay. I love company. Somebody else, they haven't heard the bad jokes. So bacterial DNA has a single circular strand of double-stranded DNA, okay, that's continuous and then supercoiled, as opposed to our nuclear DNA, which is in pairs. Okay, 23andMe thing. That's number one. The other thing is our ribosomes intracellularly, whether they are free ribosomes or bound ribosomes, based on their Svedberg units or ADS. There's a small and large subunits together, the density factor. Whereas in mitochondria, there's 70S, which are prokaryotic ribosomes. That's a better justification of evolution than what Darwin did with the Galapagos Islands. Okay? See, you learn something new every day. Goodbye. Have fun. See you Monday. I don't know why I do. Why do I don't deserve this abuse.